Hello, welcome to the Cambridge University Herbarium. My name is Lauren Gardiner and I'm the Curator of the University Herbarium, and today I'm ably assisted by Kathy from the Sainsbury Laboratory in Cambridge, where the herbarium is located. So before we go any further, I'm going to take you through into the herbarium and I'm going to tell you quite a bit more about this gentleman, John Stevens Henslow, who we consider to be our modern founder of the herbarium. So we go through now, and Kathy is going to follow me at a social distance. So this is the prep room for the herbarium. This is where we have a large freezer where we can freeze all material that comes into the collection. That keeps pests out, and it means that we don't have to use lots of pesticides and dangerous chemicals to protect the specimens. But I can tell you more about that, and you can ask questions in the chat. We have two large workrooms and a camera room, which hopefully I'll have a chance to show you as well. Kathy's going to do a little scan around the room just to show you the space, but then I'm going to go into the herbarium proper and show you some of the collections. Okay, so this is where the herbarium collections are actually stored, and it's a pretty much climate controlled space. We keep specimens cool, we keep them dry, we keep pests out as best we can to stabilise the specimens and keep them protected for hundreds of years. This space is in the basement of the Sainsbury Laboratory. Sainsbury Laboratory is on the Botanic Garden site, but the herbarium itself is actually part of the Department of Plant Sciences. So Kathy is just going to show you a little bit of the layout of the room, and you can see we've got compactor units, and these are movable storage with shelves filled with specimens. Round the outside we've got drawers, and the drawers contain packets of specimens. So these are usually smaller specimens, things like mosses, liverworts, lichens, and these are all arranged systematically within the drawers.
So if we walk down the main aisle of the herbarium, on the left-hand side the compactors are filled with collections from all over the world, and a lot of this material is from the 19th century. On the right-hand side, we have the continental European and the British and Irish collections. And here's a section of the British and Irish collection that's open, and you can see some of the specimens inside. If we walk a little bit further down the world collection side, another of the compactors is open, and again you can see more specimens laid out in folders systematically within those shelves. A lot of the material is laid out systematically, but there is still material that's in boxes that hasn't been incorporated, and this is a bit more difficult to find specimens within. And although the herbarium doesn't have a proper database yet, because everything's laid out in such an organised fashion, you can generally find things manually if you know the plant name and where in the world it came from.
Now, the herbarium was founded in Cambridge in the 1760s with the collection of John Martin, who was our second professor of botany, which was donated to the university. And that collection, part of it still remains. We have some of it here, and I'm going to show you a few specimens from there. This is part of this Martin collection. So this is from the 18th century. Now, most of the Martin collections are from the early 18th century. So the earliest specimen that I have a date on is 1703. These are specimens which were collected before Linnaean taxonomy was devised and popularised. So these have long Latin names on them, and many of them later on have the Latin binomials, the Latin genus and species name added onto them. But a lot of this material has never really been studied, and this is a collection that, as a matter of urgency, I am working on making accessible for researchers to use. Now, as I said, the collection started in the 1700s, but it wasn't until 1825 when John Stevens Henslow, who many of you may know the name from the Botanic Garden in Cambridge, became the Professor of Botany in Cambridge, and he inherited John Martin's collection, and he added a lot of specimens to it, and he recurated and salvaged as much of that material as possible. This is one of Henslow's specimens, and you see this specimen has multiple plants which Henslow collected, all are actually really interestingly arranged. He has a very distinctive way of collecting and mounting his specimens. We've got all our different plants here, different sizes, different growth forms, but then down at the bottom, Henslow very systematically added the name of the plant, the location of the plant where it was collected, his name and the date it was collected on, and he was very systematic and methodical at doing this. These are incredibly useful specimens because they tell us that what was growing at a very particular place at a very particular time. Increasingly, these specimens are used in conservation work. I used to do quite a bit of that kind of work myself, and these specimens are really valuable biodiversity data. Here's another one. This is a really lovely one. This specimen was collected with Leonard Jennings. Leonard Jennings' entomological collections are in the Museum of Zoology in Cambridge. And we have a project with zoology linking up our collections and exploring the data that these specimens reveal. So Henslow basically resurrected the 18th century herbarium, added enormously to it, did a huge amount of research himself, but was really influential in teaching botany in Cambridge. His specimens he used in his teaching. He added illustrations to lots of them. This is one where he's taken an illustration from a publication and he has actually annotated the illustration. So this is Henslow's handwriting on a hand-coloured plate from a publication. And he's added the names of all the different parts, and then he's added some of his own material, including the usual form of this bud on this Californian poppy, but then also the monstrous calyx, as he calls it. This is the bud which is dividing strangely. So this is a mutant, and that's one of the areas of research that Henslow was particularly interested in. But we know that he used these specimens not only in his research, but in his teaching as well. He illustrated a lot of specimens himself. Here I have, in the next folder, some lovely illustrations by Henslow. This folder here. Actually, here's a sheet where, again, he did a lot of cut and paste from publications. So these are other people's illustrations on this sheet, showing all the different parts of the plant. But then on the, oh, there you are. Can you see that okay? So these are really lovely, and there's records that Henslow passed these around during his lectures for students to study as he was talking about the plants themselves. But then there's things like these watercolours. These are watercolours by Henslow of fungi, and these are all collected in the Cambridgeshire area. So we've got Gog Hills, we've got Bottisham Common, and these are from the 1820s to 1830s. He was an incredibly talented man, and as I said, very influential in teaching of botany. Now, one of his most famous students is Charles Darwin, and Charles Darwin's collections are here as well. So the main collections of plant specimens collected by Charles Darwin came to Henslow, and they are in the Cambridge University Herbarium. So here's one of the specimens that Darwin collected on the voyage of the Beagle. I could tell you a lot more about these. I could tell you a lot more about Henslow and how Darwin ended up on the voyage of the Beagle, but I don't think we have time, unfortunately, today. But here you can see we have a grass specimen that Darwin collected on the voyage of the Beagle, and he has collected multiple representatives of what at the time he thought were the same species. Subsequently, they have actually been identified as two different species growing in the same place, but he collects in a very similar way to Henslow, where he's collected the same plant growing in slightly different ways, and he's documented that all together. And then Henslow, who actually put this specimen sheet together, he's mounted them together in the same way that he mounted his own specimens. Now, these specimens are really important. These are some of our most important specimens because they are so rare. There's so little Darwin material, and it's about 1,000 sheets. We have a lovely specimen, and this is a fern, and this is from the Galapagos Islands. So this is a fern specimen where so this was collected from the same plant. This is the same fern, but you see this one is a completely different form, and this is the mutant. This is a mutated form where instead of having all of the leaflets arranged like a feather, a pinnate arrangement, they are more palmate. They're all coming from the same point. And so Darwin was very interested in observing these differences, documenting what he saw, exactly in the way that Henslow taught his students to do. One of our other really important sets of specimens is our type specimens. The type specimens are usually the original plant material that a plant name is attached to. So when you discover a new species, you make a specimen and you deposit it in a herbarium like this one. And that becomes the type that name is always attached to. So for researchers, these are some of the most important collections that a herbarium can keep. And we have a very rich type collection. Here is an example of one of our types, which has an illustration attached. And this is a type specimen collected by John Lindley. John Lindley was a very influential man, very important botanist. He documented a huge number of new species, and so a lot of his specimens are type specimens, and a lot of them have illustrations, either the original piece of artwork or a hand-coloured plate, which was produced when he described the species. Here we've got a little hand illustration by Lindley on this plate. So this is one of the original hand-coloured plates done of this particular iris. Now, these specimens are incredibly important. Most of them haven't been documented, and most of them haven't been made available yet for people to research, but of all the people who come to visit the herbarium in Cambridge, the Lindley specimens are one of the most important collections for people to see. Here's another one. This is a rhododendron. This is a specimen from Borneo, and it's a large rhododendron, Brookianum. And this is on several sheets, and it has two original pieces of artwork on it, showing two different colour forms. So if I lift this one up, so here we have leaf material and large flowers. There's a packet with some extra material in, so that this material is easier to put under the microscope. You can even get DNA out of some of these early specimens nowadays, but we have to be careful about how much material we use because that's a destructive process. And then here's one where we've got the red form. This is the original piece of artwork. And there's a whole area of research just studying these illustrations. Who did them? When were they done? Why were they done? And then this is the yellow form, which was documented. I haven't told you numbers. I've told you you've got a lot of material. I've shown you lots of things on the shelves. But this is a major collection. We have about 1.1 million specimens estimated in this collection. That's one of the biggest collections there is. We're within the top 50 herbaria in the world in terms of size. There are well over 3,000 herbaria worldwide, and we are fourth largest in the UK. The UK is very rich for natural history collections, and Royal Botanic Gardens Kew, Natural History Museum in London, and Royal Botanic Gardens Edinburgh have the three largest in the country. Those are huge, huge collections. We're next. So this is a major collection. It is very important, and it urgently needs more research to be done on it. I mentioned conservation, but there's a lot of research, modern research, which is done with these kinds of specimens. And unfortunately, this collection hasn't been used very much in recent decades. It has a very, very rich British collection. Our British herbarium is enormous, and is one of the most significant ones in the world for Great Britain and Ireland. We have very extensive continental Europe collections, but then also these world collections, specimens from the tropics, particularly things which were collected in the 19th century, very, very rich. And a lot of that material is type specimen. So there's a lot here and a lot to do. Currently it's only me working here. I'm the only curator. And the plan is hopefully to expand our capacity in the future so that we can do a lot more research and we can embed the collection back into the teaching of the department and the wider university and generate a lot more research out of these collections. A really key part of that work is digitising our specimens. It's imaging these specimens, taking high resolution photographs of them, databasing them and making them freely accessible online. So I'm just going to show you next door the camera room. This is our digitalization suite, where we have a setup that allows us to do this. And I will take one of these specimens with me to show you how we do that. Okay, so this is the digitization room. We have a herb scan. This is a high resolution scanner, specially adapted to scan specimens. And this is on loan from the Royal Botanic Gardens Kew. They very kindly helped us out by lending us one of their scanners. Here is one of our specimens. You basically place the specimen on this frame. It's on springs, it's on sponge, and you place the specimen there. You raise it up very carefully, and because you're doing it carefully and you're not turning the specimen over, you protect the specimen and hopefully do not damage it. It's pretty essential. And you essentially scan the specimen. This is a workhorse of many herbaria around the world. A huge number of specimens have been digitized using these. But we were able to use this to make a pitch to the university, to the Department of Plant Sciences, to get a high resolution camera and light box. If I turn the light box on, you can see we have a nice high brightness set of lights. We can put our specimen directly underneath and we can take really beautiful high resolution photographs straight down. And we have a scale bar, we have colour charts, very, very standardised. And now we can make these specimens available for people to use. Currently, mostly I try to service requests. So when somebody is doing a particular piece of work, say on iris, they will request images of material. But the plan is to digitize the whole collection ultimately, but to prioritize our most important collections and to image them all together, make them available and accessible online. And hopefully by the end of this year, we will have our first collections properly online again. And we can start really making headway with this research. So I hope that has been an interesting overview, an insight into the world of this herbarium and herbaria in general. We're now going to play a short video. It's an interview that I did last year with BBC Look East, which shows you a little bit about some of the things I've just shown you, but a little bit more, including one of the Darwin specimens which we found in the collection, which had previously not been documented. After that, we're going to do some questions and answers in a couple of minutes, and in the meantime, the video will be played. Thank you.
Some of these files haven't seen the light of day for 300 years. This basement is home to more than 1 million plant and fungi specimens, collected from all over the world by some of our most famous scientists. You found this at the back of a cupboard?
Yes. Yes,
like you do.
Like you do.
And what exactly is it?
So this is a fungus specimen that Darwin collected on the voyage of the Beagle, and he collected it in Tierra del Fuego in South America. It's a species which is still found there, and local people eat it.
Well, it looks gorgeous. I won't try it now. In fact, it's what's known as a type specimen, which means it's the original one they used to name the species. Lauren's been curator here for just over a year. Before that, many of these specimens lay virtually untouched.
Pretty much every day that I work in the collection, I discover new extraordinary specimens. So this is the piece that I discovered most recently that is my favourite, and I really love this piece of artwork, and this is original. This is a relative of hydrangeas, and this plant was collected by Robert Fortune in Hong Kong. So here you've got these ridges, and they actually shimmer gold still on this specimen, which I think is quite beautiful. The insects would slip down into the pitcher and be digested inside. So this specimen is of the tea plant Camellia sinensis, and Darwin collected this on the voyage of the Beagle in the Botanic Garden in Rio de Janeiro.
It might be 3 centuries old, but this collection is vital for modern scientists who want to understand how climate change and urbanisation are buffeting the natural world.
These specimens tell us how plants have changed over more than 300 years. We can look at how the environment has changed, how the climate has changed, how humans have impacted on plants in the wild, and we can use that to predict and plan for the future as well.
So who knows what other treasures remain hidden away down here? Extinct species, original specimens. Lauren says she's barely scratched the surface.
I find the detective work of piecing together the story, making sure that it is that original specimen, a lot of fun. It's also quite a privilege to be handling this material.
Richard Westcott, BBC Look East, the Cambridge University Herbarium.
Hello, so thank you very much, everybody. I hope that was interesting. I've got lots of questions in the chat, which I am going to work through now. This is from Irina. Thank you for showing us the collections. It is all very beautiful and interesting. You mentioned an urgency to making the samples accessible. Where is the particular urgency from? So for the kind of research that I specialise in, it's conservation, and conservation, as many of you will know, is pretty urgently needed for many species and many different areas around the world. These specimens are the fundamental data that underpins a lot of research and conservation work, including IUCN Red List conservation assessments. So when we're trying to prioritise which species to focus funding and conservation efforts on, we assess species and we also assess different vegetation types, habitat types, different locations. The species conservation assessments are often, in particular with plants, often based on herbarium specimens. So every single herbarium specimen around the world shows you that a plant was growing at a particular place at a particular date, and often more information about it, associated species, vegetation types, uses, human communities, etc. If you have one specimen, that's great. If you have 50 specimens of the same species from different locations, 100 specimens, 1,000 specimens, you have a huge data set there which allows you to know how things have changed or predict how things might change in the future. That work is pretty urgent. We need to be figuring out what we need to concentrate our efforts on. And a first step towards that is digitizing these kinds of collections and making them available for researchers to feed into that work. So that's just one area of research. In terms of species description, a lot of specimens documented from all over the world, so in the room behind me, all these specimens, they tell you what was growing where and when. Some of the specimens in this collection are extinct in the wild. We haven't found them since. There's lots of research, there's some great research which came out of Oxford University over the last few years, where they studied how many of the specimens in their collection have never been described before, and they discovered a huge number of new species just in collections rather than being discovered in the field right now. That allows you to target your research, to go out and look for things and find out what's happening. And all sorts of other areas of research. So yeah, there's a lot of work going on. That's really skimming the surface though. Digitization or digitizing must be very time consuming. Do you have access to a technician? This is a question from Anne Loveday. Thank you very much for this question too. Yes, it can be quite time consuming. Unfortunately, at the moment, we don't have a technician. Again, as I mentioned, our plan is to be fundraising to support this collection much more in the future, to open it up, to make it more accessible, to have more research going on, and to get more teaching using this collection, because it is a really fantastic resource that we're not making full advantage of right now. My plan is hopefully to be able to resource a technician in the future. I had a short-term contract for a technician earlier this year, and we were able to start digitizing some nice chunks of collections, including that Martin collection. So over the summer, kind of around the lockdown period, we were able to have that technician image all of the 18th century material, and the plan is for that to be one of the first collections that is made available online. So please watch this space. There will be some publicity around that when we get to that point, but hopefully that will be soon. The digitization now with the camera, with the herb scan, which is the scanner which is mounted upside down, and now with the camera, digitization is much quicker than it used to be. So we can do hundreds of specimens in a day. We've basically taken high resolution images, and we can make those available, and we can digitize the content, the data from those images. And collections are increasingly using virtual volunteers and staff off-site to be transcribing that data from the images. That is a really fantastic activity for people to engage with the collection around the world, but also during times when they cannot physically access the collection. And so lots of herbaria and also natural history museums, zoological collections, geological collections have been doing exactly this. They have been making lots of the images that they have of their specimens available and getting members of the public to basically transcribe the data from them into their databases. So the plan is to start doing some more of that. And I've done a little bit of piloting, again with the Museum of Zoology over this summer with a small group of students trying that out for the first time for us. So it can be time consuming, but there are ways of expediting that kind of work. Right, next question. I'll just mark these as done as I go through. According to what system is your herbarium sorted? Do you feel secure that future specimens will fit into the herbarium, i.e. do you have enough space? This is a question from Mariette, and those are two excellent questions. So our collection is arranged by several different systems, which does make it a little bit more difficult sometimes to use, but as long as you know the system, you can find things. It just might take a little while sometimes. So the collection, which is Great Britain and Ireland, that is arranged according to a particular flora, a set of books, essentially, which were written actually in this collection. And they were written by Peter Sell and Gina Morel. A really, really comprehensive flora of Great Britain and Ireland. And if you use that set of books, the specimens here are laid out in exactly the same sequence. So you can use that as an index to that part of the collection. The world collection, which most of it is much earlier, it's got a lot of material from the 19th century. That is arranged according to the Bentham and Hooker scheme. So that's a big flora of the world. Some people ask, are you going to rearrange the specimens? Currently, there's no urgency at all to do that. As long as you know which system each section is laid out in, you can find the material. The more important thing is to start databasing the material so that we can search it much more easily. I sometimes describe it a bit like a library system. So you might have a library arranged by Dewey Decimal, or a library arranged alphabetically, or by different themes. As long as you know the system, you can find the material. Unfortunately, because we've got a lot of material that hasn't been processed, hasn't been arranged, and we've got multiple systems, it can be more difficult, much more difficult. Regarding the space issue, we do have some expansion space, as we call it, in the collection. Some herbaria, they accession a lot of new material every year. Kew, for example, adds about 25 to 30,000 specimens every year to its collection. That's from their own fieldwork, from teams of researchers working around the world with collaborators, collecting material and putting it into the collection. Also swaps. We swap a lot of material, we exchange and we gift material to different collections. The Cambridge University Herbarium doesn't actively add a huge amount to the collection, but we do add several thousand specimens a year, mostly in the form of requests and gifts from individuals. We're not adding an enormous amount of material, but we do have enough to keep us going enough space to keep us going for a while. But if we were to become a collection that was actively adding a lot more material, then we might have problems because we don't have a huge amount of expansion space. There is a question about sugar beet from Anon. So do you have any old sugar beet samples in your collection? If so, could you please elaborate on samples, collection dates, etc.? I do sugar beet research and therefore are highly interested in beets. Thank you, and thank you for the compliment that you've added at the end of that question. I don't know, we probably do. Unfortunately, because we don't have all the collections database, I can't do a simple search. But to answer that question, I would look up, make sure I know the taxonomic name of a particular thing somebody is interested in. Unfortunately, I have to look up all of the synonyms, all of the older names, for historic material, which might still be laid out under old names. And I'd go to the right parts of the collection where that material should be, if we have it, and I would physically look. So I'm afraid I do not know. I hope we have some sugar beet material, but I can't tell you, unfortunately, right now. That's a really good example of the kind of query I get a lot of, and unfortunately, I just do not have capacity to look for every single thing and to be able to answer people really quickly on such things. There's another question from an anonymous attendee. How did early collectors prepare the specimens? Do you notice any differences between how well specimens have been preserved between collectors? And have you found anything interesting that was what the collector intended to preserve? Brilliant question. Lots of people ask me this, and they ask me how I make specimens now, how botanists make specimens in the field. The techniques are broadly the same. We essentially press plants. So if you think of pressing flowers, we do pretty much that, but with whole plants or set big chunks of plants. We use big herbarium presses. They're giant flower presses, and we use newspaper and sometimes blotting paper and other materials to absorb moisture. We basically build up a sandwich. So you have your press, and then you have layers of absorbent material and newspaper and your specimens all sandwiched together inside a press and then strapped firmly together. And ideally you can add dry heat, gentle heat to your press so that the moisture is carried out of the specimens, and you get flatter and flatter, which helps for storage as well. And it preserves them before they start to rot, and that's very important. If you dry your specimens well, then you can preserve, and then you keep them dry, keep the pests out, keep them cool, you can keep them for a long time. So the way that we make specimens now is pretty much the same as early collectors made them, and the earliest specimens in collections around the world go back 400 plus years. So it's very, very similar. There are slightly different ways and different techniques. There are different things that people do, but that's broadly the same principles. Older material can start to degrade, so some of the early material does have signs of having been damaged by pests or going mouldy, but we have specimens here which are 200 years old, which still have bright blue flowers or bright purple red flowers on them. So it can be quite variable. If they were dried really well in the first place, quickly, but not too quickly, not cooked, not scorched, not allowed to rot, and they've been preserved well ever since, then they can be incredibly fresh looking. Thank you. Next question, I've answered the one about how much new material is coming into the collection. Thank you, Marianne, for that question. Who sends new material to our herbarium? So we do have some new material coming in, and most of that new material is from gifts, so people who are leaving us their collections. We do need a bit more of a formal accessions policy of what we do keep and what we don't keep. If there's a collection that somebody wants to leave us and it's not something that we really have space for, or the skills or the expertise here, or the particular collection's knowledge and need to add, then I will always refer people to other collections around the country, where that material might find a better home. But ultimately, we try to never throw anything away, because some material that you might not think of as being particularly useful, many duplicates of the same species from the same place, actually that is incredibly valuable material. But if you don't have space to store it and the resources to store it, then it becomes quite difficult. You mentioned that all specimens were frozen when entering the herbarium to destroy pests. Does this not damage the specimens? That's a brilliant question, Sue. So we make sure that all material is completely dry before we freeze it. If it is not, then yes, you will definitely damage the specimens. A bit like freezing strawberries. They turn to mush when they defrost, and then actually that will accelerate them going mouldy and degrading. So as long as the material is really well dried, we can freeze it. We freeze it at -30 to -40 degrees for at least 3 days, and then we bring it back up to room temperature. But the specimens are sealed in usually in boxes, but then in plastic bags, so that any moisture that was in that environment, then when it defrosts, you don't have condensation forming. Because again, that basically makes your specimens wet again, which will accelerate their decomposition. So if you do it well, if you do it properly, you bring the specimens back up to temperature in that closed space, then you accession them and put them away in the collection. Then they do survive very well. You don't want to really be freezing specimens repeatedly. So when we do take specimens out of the collection, before they come back in, they are refrozen. And if you did that very, very frequently, then yes, you do risk damaging them. But if you freeze them on the way in, and then you use them in this clean space, so we keep this as clean as possible, we keep as many other all plants out, we make sure that if people have muddy shoes, they take them off, or we have shoe covers. We try to keep it as clean as possible in here. And then we shouldn't need to keep refreezing. If we do have an outbreak of pests, and we do monitor for pests, because they will eat the specimens and damage the specimens, then we might have to use some chemicals. There are only a few that we are able to use nowadays under strict controls. We clean the spaces, and we might refreeze sections of the collection to stop that pest spreading and damaging more material. Okay. What is the difference between a specimen in general and a type specimen? That's a question from Fangda. Thank you for that question too. So all of the collections are specimens. The type specimens are the ones that names are attached to, like in terms of when you describe a species, a type specimen is designated. So it's a specimen which is the one that name is sort of permanently attached to, a bit like a dictionary entry, which is the specimen. There might be some duplicates of those specimens. So if I discover a new species in the field, on a field trip, I make some specimens from that one plant. I will make one specimen of what we call the hollow type, that is number 1 type, and that gets deposited in a herbarium as the holotype. Any other material that was collected from the same plant at the same time, those are kind of secondary types. They're part of that type collection, that original material for that name, but those we call isotypes, and we send those isotypes to other collections around the world so that there's a copy somewhere else. It has to be from the same plant, it has to be from the exact same time, the same gathering. But that's what the types are. They're a subset of specimens. Prints. So this is an excellent question. So this is from Amanda. Prints. Where may one purchase prints of the things you showed? Currently you can't, but hopefully in the future we'll be able to do that. And I'm talking to the University of Cambridge Fitzwilliam Museum Enterprises about the possibility of licensing some of our images and having some merchandise and some prints that people can buy, because people do love them. I love them. I have some copies which I produce myself, and the walls in my office. But yes, I hope that in the future, if you do like this material, you could always email such people, contact the Fitzwilliam Museum Enterprises and say that you'd love to buy things like that, or tweet us. Yes, I hope that we'll be able to do that in the future too. Oh, this is a question from Hike. Hello, Hike used to work here in the Sainsbury laboratory. Thank you very much for this. Thank you. Is there a connection between the Cambridge Herbarium and the Cambridge Philosophical Society? Was it used in debates or talks presented by the Society? Maybe even in debates on Darwin's On the Origin performed within the scope of the Society? Yes, so John Stevens Henslow was one of the founders of the Cambridge Philosophical Society, absolutely. And his collections, his botanical collections, are here in the herbarium. His zoological collections are in the Museum of Zoology. Their geological specimens are in the Sedgwick Museum. Absolutely, this is essentially the botanical outpost of the Cambridge Philosophical Society, for sure. It would be fantastic and fascinating to have a researcher actually track back and see how material was used by the Cambridge Philosophical Society. But the material that Darwin sent back from the voyage of the Beagle, Henslow published some of the letters that Darwin was writing to him, accompanying those specimens. He published those in the transactions of the Cambridge Philosophical Society. So absolutely they are entwined, and that's another connection that hasn't really been opened up, research, brought to light, and I hope that we'll be able to do that as well in the future. Oh, another excellent question from Fang Da. I hope I'm pronouncing your name correctly. What is the difference between a herbarium and a botanic garden? So a botanic garden is living material broadly and usually openly accessible, publicly accessible. A herbarium is a preserved collection, usually of dried material, but some collections have material in spirit, in pots of alcohol to maintain three-dimensional structures. A botanic garden can have a herbarium, but a herbarium doesn't always have a botanic garden. We are lucky enough to have both here. So the Cambridge University Herbarium, which is part of the Department of Plant Sciences, that is the collection that I am responsible for, and it's here. The Botanic Garden, which is above me and out adjacent to the Sainsbury Laboratory building, they have their own herbarium, and that's actually stored in this space as well, and that's a small collection, but a very important collection as well. Often a Botanic Garden's herbarium will contain cultivated material from that collection, sometimes exclusively from that Botanic Garden. We've got a mixture here, so the Botanic Garden does a lot of active research and they collect specimens on their field trips, and then they make specimens of their own collections in the garden, and that material goes into their herbarium. But we also have a lot of material from the Botanic Garden that is in the main University Herbarium too. And so there's a bit of a blurred line there, but because they're on the same place and we're all connected, we can access the material and research it together. The Botanic Garden here has actually databased all of its collection. So that is something that we can search much more easily than the main university collection. Are there any rare and unusual and extinct species in the collection? Secondly, as well as the evident historical interest, has there been more scientific or medical research done on rare items in the collection? This is another anonymous question. Excellent questions. Yes, definitely. We definitely have specimens here of rare and even extinct species. Again, I can't tell you how much because a lot of it has never actually been researched in modern times, but I know of at least one Darwin specimen which is extinct in the wild as far as we know. It's never been seen again since, and we have the only specimen of it in existence here. So, you know, that just highlights how important these collections are in some ways. If that specimen hadn't been made by Darwin, and if it hadn't been preserved and we didn't have it anymore, we would never know that species had ever existed. And there was some interesting research done with that one where they were able to take some DNA from it and to check whether or not it was closely related or the same species to other things. And it made it very clear that no, this was a different species and it hasn't been seen again since. But there will be more material like that, unfortunately, in the collection. And there will be some very rare things. In terms of historical versus modern research, this collection is very, very rich in historic material. But that is not to say that we do not have modern material too. We still have modern material that was collected within the last year in this collection. And we have a huge amount of 20th century and early 21st century material too, particularly the British collection. There's a lot of British material from the last 100 years. There is a lot of research that could be done with the material, and some research institutions, herbaria, they do research that is medicinal, looking at medicinals from species around the world. Crop wild relatives is another really interesting and important area of research. So if you know a particular plant has useful characteristics, either for food or for medicinal uses, you can figure out what the relatives are of it, and you can basically look in these collections and you can study whether or not they have the same properties, better properties, higher productivity perhaps. So these are all really, really useful, current, modern areas of research. If you think of a herbarium as an enormous repository of fieldwork, essentially, you can go do fieldwork now and you can be looking for new medicinals, new drugs. You can look for and work with communities about different traditional uses and different foods. And you can look to find crop wild relatives. And you can make specimens now for that kind of field work, but you can also go to these collections, and this is basically other people's field work in the past, all documented here for people to research. So there is a lot of research around the world which is done with herbaria. Literally, if you just Google herbarium and research, if you look at some of those big collections I mentioned, Natural History Museum in London, Royal Botanic Gardens Kew, Royal Botanic Gardens Edinburgh, and you find out about the kind of research that they do with their work, with their specimens, you'll find out a lot more about the kind of things that people are doing around the world. And I hope that we will be doing a lot more of that with this collection in the future. Okay, we have 5 minutes left, so I will have to wrap up soon. I have a huge number of questions still to answer, unfortunately. I'll try to skim through a few of them, but hopefully I can answer some later on, maybe when this goes on to the YouTube channel. Do you have a seed bank? Asked Simon. We don't for this herbarium. The Botanic Gardens in Cambridge does have a seed bank, but no, we don't specifically here for this herbarium. Are you involved at all in changes in nomenclature of plants? This is another question from Anne. I certainly have been in the past, and yes, we are. A lot of researchers who use this kind of material, they are revising the taxonomy, the classification, and the names of plants. Absolutely, really fundamental science for all plant research and conservation too. And in my previous life, I worked at Kew and I was a botanist on the staff. And yes, I was involved in DNA work, molecular work, trying to work out how things are related and renaming species, reassigning things to different groups. Zoe asks, how many samples do you have from New Zealand? I can't tell you because I literally have to go next door and count them. That is something that a proper digitization program and databasing program would allow us to be able to answer in the future. Currently, we'd literally have to have somebody come around and count specimens, and because they're in lots of different locations and we've got a lot of material that's never been actually researched, that's going to take a little while before we're at the point where we can do that. Mariette asked a similar question, how many specimens are not determined even yet to genus? We call these indeterminate species. A lot. Again, I can't tell you, but yeah, we have plenty of material that has never actually been identified properly, or maybe if we look at it now, we study it now, we'd actually find that the names have changed, or we have got a new species, etc. So I'm afraid that's a really unhelpful answer, but we will have a lot. I think I've answered Brian's question, what is the most rare specimen you hold? That would be the Darwin specimen I mentioned. It's called Sikios villosa. Oh, because I haven't moved for a while, all the lights have just gone up, so I'm just going to move. Motion detectors on the lights. Thank you, Caroline, for your very kind comments. Good luck with the funding. Yes, thank you. Please do watch this space for more information. And thank you. Oh gosh, there's a lot of comments which are lovely thank yous. Is there any scope for taking DNA samples of extinct or endangered species and reproducing them? If so, how is that done and where is the DNA extracted from? That's a question from Irina. So increasingly we can get DNA out of old specimens with different techniques, modern techniques, and that's a lot easier than it was even 5 or 10 years ago. It used to be that older material, it would have degraded so much that the DNA, you couldn't isolate it, you couldn't extract it and sequence it. But increasingly we'd be able to get DNA out of old material up to 200 years or more. Still not easy though. In terms of reproducing them, theoretically, maybe that is something that people could do, but resources are finite, and actually we probably, in my personal opinion, I know a lot of researchers and botanists feel the same way, it is better to spend the money we have on conserving what we still have, and that has urgent need to be conserved, rather than try to resurrect things. But yes, theoretically we can get DNA increasingly from old specimens. It means we can study things, we can work out how things have changed, how things may have evolved on even relatively short time scales. So Richard asked a similar question about DNA, but has anything interesting emerged from analysis of DNA from your specimens? Yes, I think that the Darwin extinct specimen is a nice example of that, that the DNA from that specimen showed very distinctly it was a different species, that it was not related to the other species geographically close, and that yes, this was something unique. Thank you very much. These are excellent questions. I think we are going to have to wrap up now. Yeah, thank you. I'm just seeing if there's anything I could answer quickly. There's questions about mutation. That is a whole area of research, and I do not have time to go into that. But yes, there's lots of genetic mutations. If you get lots of specimens, you see the the normal state of species. You can see the variation that you see. You can pick up some of the mutations. So that's again why it's really valuable to have lots of collections in our collection, but in collections around the world, so you can compare and you can see how things vary worldwide. So thank you very much, everybody, and thank you for all your kind comments. I hope this has been interesting, and I look forward to telling you lots more about the herbarium in the future. Hopefully we will have a new website and we'll be getting our specimens available online over the next year or so. But by the end of this year, there'll be more news. So I'm going to sign off now. Thank you again and enjoy the rest of the festival. Goodbye. Stay well.